How does the human body coordinate complex physiological instructions across vast distances when it cannot rely on rapid electrical impulses from the nervous system? It relies on a specialized chemical network. The functional unit of this network is the gland, a biological factory designed specifically to synthesize and release precise chemical substances. There are two distinct anatomical categories for these factories. The first is the exocrine gland. Exocrine architecture is defined by the presence of physical tubes, or ducts. Chemical catalysts manufactured here, like digestive enzymes or sweat, travel through these channels to be deposited directly onto specific epithelial surfaces or into particular body cavities. Because these chemicals must physically travel through a pipe to reach their destination, their physiological reach is strictly confined to the immediate area where the duct opens. Therefore, exocrine systems are built to exert immediate local control over a specific microenvironment. To achieve systemic regulation, the body utilizes an entirely different structure, the endocrine gland. The defining characteristic of endocrine glands is that they are completely devoid of ducts. Instead of relying on dedicated pipes, these cells secrete their chemical messengers, hormones, directly into the surrounding interstitial fluid, where they are quickly swept into the broader systemic circulation. This vascular access grants endocrine hormones a massive operational range, allowing a single gland in the brain or neck to alter target cells anywhere in the entire body. Biological rules, however, often contain exceptions. Some organs possess both sets simultaneously. The pancreas is the primary example. It contains exocrine acinar cells that secrete digestive enzymes through localized ducts, sitting alongside endocrine islets of Langerhans, which secrete insulin straight into the bloodstream. Whether a gland utilizes a targeted duct or taps into the global bloodstream, its macroscopic physical architecture strictly dictates its physiological reach. Once a hormone enters the bloodstream, the challenge shifts from how the message is dispatched to how the target cell actually receives it. Every target cell is protected by a formidable barrier. The cell membrane is a lipid bilayer, a highly selective, fat-based perimeter guarding the intracellular environment. Many chemical messengers, such as peptide and protein hormones, are water-soluble. Because water and lipids repel each other, these water-soluble hormones are physically repelled by the cell membrane. They cannot breach the exterior to deliver their instructions inside. The structural workaround requires an intermediary. The peptide hormone approaches the cell and locks into a matching surface receptor on the outside of the membrane. The stocking activates an internal enzyme, converting ATP into cyclic AMP inside the cell. Cyclic AMP acts as the true second messenger. It executes rapid physiological changes, like opening calcium channels, while the original hormone remains locked safely outside. Peptide hormones trade deep cellular access for explosive, high-speed physiological reactions that execute in fractions of a second. This surface-level interaction stands in contrast to a different class of messengers, steroid and certain amino acid hormones. Their defining chemical trait is that they are lipid-soluble, melting straight through the cell's fatty exterior area. They bypass the need for external surface receptors. They cross the cytoplasm unimpeded to breach the nucleus itself. Here, they employ a gene-specific mechanism. The hormone binds directly to the DNA, triggering the transcription of a specific messenger RNA sequence. That mRNA exits the nucleus and is translated into brand new functional proteins, permanently altering the internal machinery of the cell. The physiological cost of this deep infiltration is time. Building proteins from scratch is a slow process, often requiring hours, days, or months to complete. However, the resulting biological changes are highly sustained and deeply structural. A hormone's core molecular solubility determines whether a target cell will react in seconds or slowly adapt over the course of months. Moving back from these localized cellular mechanics, we have to look at the macroanatomy to understand where these chemical orders originate. The apex of this control hierarchy is the hypothalamus, a neuroendocrine bridge that translates raw neurological processing into tangible chemical outputs. By synthesizing specific releasing and inhibiting hormones, the hypothalamus governs the entire systemic network. It issues these commands directly to its primary executioner, the pituitary gland. The pituitary is divided into two distinct regions, starting with the posterior lobe, or neurohypophysis. The posterior lobe synthesizes absolutely zero hormones of its own. It serves entirely as a storage tank, holding and releasing hypothalamic hormones like ADH and oxytocin. This strictly structural role contrasts sharply with the anterior pituitary, or adenohypophysis, which is an active chemical factory. The anterior lobe translates signals from the hypothalamus by actively synthesizing its own library of chemicals. It produces tropic hormones that command other downstream endocrine glands and non-tropic hormones that target peripheral tissues directly. Maintaining strict systemic homeostasis requires this rigid, top-down anatomical command structure. When a single node in this tightly regulated hierarchy misfires, the systemic consequences are severe. Consider a specific failure at the anterior pituitary, the chronic overproduction of a non-tropic hormone known as growth hormone. The physiological outcome of this error depends entirely on developmental timing. If hypersecretion occurs during childhood, before the epiphyseal plates in the bones have closed, the excess hormone drives extreme linear bone growth. This pathology is known as gigantism, resulting in the rapid, uncontrolled lengthening of the limbs and a massive final vertical height. The exact same chemical hypersecretion presents completely differently if it occurs in adulthood, after the skeletal growth plates have permanently fused. When adult bones are flooded with excess growth hormone, they can no longer lengthen. Instead, the skeletal structure reacts by expanding outward, thickening the existing bone matrix. This adult-onset disorder is acromegaly, characterized by heavy, thickened features in the jaw, the brow ridge, and the extremities. Decoding these extreme clinical manifestations in medicine requires a rigorous understanding of the underlying glandular architecture.